Welcome to the Proteins and Cell Metabolism lecture. This lecture will be pretty short as well and not have many mechanisms on it. However, it is important in understanding how proteins and enzymes function. So we'll first start by talking and defining chemical work, transport work, and mechanical work. Three different types of work have the ability to make and break bonds. They can move ions, they can move molecules, or they can move entire cells, or move you as a human being. Work is required for all of those components. Chemical work makes or breaks chemical bonds, which we learned about in the last PowerPoint. Transport work, which you'll learn about in the next PowerPoint, is the work required to transport ions, organelles, molecules, salts across plasma membranes or from one location in the cell to another. Mechanical work is the physical movement of something, whether that is cilia, flagella, or your physical body in terms of muscle contractions. Mechanical work is what is used to move those components. Now, energy itself comes in two forms. Now, if you've taken physics, you know these terms: kinetic energy and potential energy. Kinetic energy is the energy exerted while in motion. So while you are running, you are exerting kinetic energy. Let's say you're running to the top of a mountain. While you're running to the top of that mountain, your body is exerting the kinetic energy required to propel you forward going uphill. Once you reach the top of the mountain, you stop and you stand still. At the top of that mountain, you now no longer are exerting kinetic energy, but rather potential energy, meaning you now have stored energy in your body because as you were hiking up that mountain, moving up against gravity allowed your body to store energy that can then be used to either run back down the mountain or roll back down the mountain. The potential energy that you or an object has is the energy that is stored within you that gives you the ability to potentially do work, not that you're actually doing work at that moment. Potential energy, once you're at the top of the mountain, can be converted into kinetic energy. Let's say you decide to lay on your side and roll down that mountain. Once you get your roll going, you do not need to continue propelling yourself forward because the potential energy that you stored when you got to the top is slowly but surely being converted into kinetic. One big rule about energy you should know is that energy is never created nor is it destroyed. So here's a very similar example of a man rolling a ball up the hill. Because he's moving as he's moving this ball up the hill, the energy he's exerting is called kinetic energy. Once the ball is up on the top of the hill, just stationary, this ball has stored energy in it, which we call potential energy. With a little bit of a nudge, the ball starts rolling back down the hill on its own, converting all that potential energy back into kinetic energy as it makes its way down. So again, with the rules of energy, energy is neither created nor destroyed. That is the first law of thermodynamics. Here, the definition that comes from your textbook reads that total amount of energy in the universe is constant. So whether you're exerting energy or utilizing it, it's being converted from one form to another. It's never truly being lost. The second law of thermodynamics states that no matter what kind of chemical reaction you have, somewhere, someplace within that chemical reaction or its surrounding environment, the entropy is always increasing. The word entropy means the amount of disorder. So the speed of the moving molecules or how crazy they're bouncing from one wall to another. A great example of increasing entropy is when you take an ice cube. In an ice cube, the molecules are pretty stationary. They're maybe like shaking in place, but they're definitely not bouncing around. As that ice cube melts into liquid, the molecules are moving faster and faster and bouncing from one end to another much faster with higher velocity. That is an increase in entropy, the disorder of the molecules. Molecules become more disordered as they gain energy or as heat is applied. Now, the next few charts you're going to see are energy diagrams. Energy diagrams can be either exergonic ones or endergonic ones. I think the key thing to see here is where the reactants start and where the products finish. On the x-axis, you have time. On the y-axis, you have the energy. The reactants start with a higher energy, and the products end with a lower energy. So because we started at a high energy and we ended up at a lower energy, we could easily say that this whole reaction lost energy. When reactions lose energy, we call those exergonic reactions. Exer, or exiting, could indicate that energy is exiting out of that reaction. Another key term to circle here is activation energy. This is the energy required to overcome before allowing the reaction to proceed forward. So if in blue you have your reactants, the reactants must increase their energy or somehow gain energy to reach the top of the mountain. That energy that you've gained is called the activation energy. And it is indicated from this point all the way to this point. So activation energy is the amount of energy we take in to get to this point. Once you've gotten to this point, you may simply roll down the hill and get to products. So with this kind of reaction, you're losing energy, and the energy you're losing is from here all the way down to here. Another type of reaction you have is an endergonic reaction, and that means your reactants have a lower energy and your products have a higher energy. The overall energy gain is right over here, from where you started to where you finished. Now again, we have activation energy, the amount of energy that we must rise up and overcome before getting to our products. Activation energy, again, is always measured from the reactants up to where that intermediate state is. This is an endergonic reaction. Energy must enter into the reaction in order to get to products. Now, what can help us make these reactions proceed faster? For example, I'm going to draw a typical graph. We have our reactants here. Let's say we have activation energy there, and we have the products down there. You need to input this much energy as the activation energy to get the reaction to actually reach the top point before it can roll down spontaneously. This is quite a bit of energy, but we have a way to lower that energy. With the use of enzymes, reactions have the ability to lower their activation energy and look more like this kind of curve. With less energy needing to be input into the reaction, do you think this reaction is going to proceed faster or proceed slower? Well, probably faster because it requires less energy to go into it. We call these enzymes catalysts. Catalysts are types of enzymes that speed up the rate of chemical reactions by lowering the activation energy. Make sure you write that down. They do it by lowering the activation energy. So, like other proteins, enzymes, which are just specialized proteins, they function optimally in specific environments. So if you notice here, the x-axis says pH, the y-axis says rate of enzyme activity. At a pH of 5 or 9, is that enzyme activity really high? Not at all. Pretty low. As you go up to 6 or down to 8, the reaction has a higher activity. But at a pH of around 7.3, we have the optimal rate of enzyme activity. That means whatever enzyme this is loves to function at a pH of about 7.3 or 7.4. If you increase the pH and make the solution more basic, or if you decrease the pH and make the solution more acidic, at that point, the rate of the enzyme activity decreases and that enzyme doesn't work as well. From the last PowerPoint, we learned about the word denaturing. When you increase or decrease pH too much, sometimes it's to such a level that acidity or basicity can actually denature your enzyme, breaking it and not allowing it to work anymore after that. All right, so slightly switching gears. All cell reactions are termed metabolic pathways. These are pathways that are chemical reactions. They are used to make ATP, to break down ATP, to digest nutrients, produce enzymes. All of those reactions are titled metabolic pathways. How do cells control those metabolic pathways? How do cells control how much enzyme they make? How well an enzyme works? How much ATP they have? Or how fast a reaction proceeds forward? Well, cells have 5 ways to regulate their metabolic pathways. Number 1, controlling your enzyme concentration. If you don't want a reaction to proceed forward, then just don't make the enzyme for it. Stop making it. And that reaction will slow down so much that it'll probably stop. Produce modulators that change reaction rates. We haven't talked about modulators in this PowerPoint, but in the last PowerPoint, you guys talked about coenzymes, allosteric activators, allosteric inhibitors. All of those are modulators that can either bind to or change the structure of your enzyme. And if you change the structure of the enzyme, you're going to change the reaction rate. Using different enzymes to make the reaction go in reverse. So sometimes when you have a reaction, and I'll write this out for you, that goes from A to B. Let's say enzyme 1 makes a reaction go from A to B. What if I made enzyme 2 then, which made it go from B to A? With the production of another enzyme that reverses my reaction, I'm able to control how much I move towards B and how much I move towards A. That's essentially regulating my cell's reaction cycles. Let's say you don't have the ability to stop making enzymes though, like how number 1 states. What else can you do? Well, compartmentalize the enzymes. Take those enzymes and store them in a storage facility so that they just stop interacting with the reaction. And the last one is maintaining optimum ratios of ATP and ADP. Sometimes reactions, and I'll put a little arrow here, require ATP, which is energy, you should know this, to help the reaction proceed forward. When you input ATP into a reaction, that's your energy source. It gets converted into ADP. So to get this reaction to move forward, you must constantly have an input of ATP. Now, what do you think you're going to need to input to reverse this reaction? To reverse it, you've got to input ADP. And what do you think it's going to make on the other end? ATP. So what this means is that when you're going in the forward direction, what do you have to input? ATP. Make sure you have enough ATP. But when you're going in the reverse direction, what do you have to input? ADP. Make sure you have enough ADP. So ATP is not just the only important molecule. ADP is equally as important. So maintaining a proper ratio of both of these guys is imperative. Now we get to the portion that I know there's a test question on, so be sure to pay attention to this next few slides, and then we'll do an example together at the end. We did touch on this on the last PowerPoint, transcription and translation, but now we're going to go into it in detail. This whole last chunk is about how you make proteins from the DNA. So, big overview. DNA is found in your nucleus. DNA can make more DNA that's identical. We call that replication. When DNA converts into RNA, we call that transcription. When RNA converts into proteins, we call that translation. Make sure you know this schematic here and the names of the processes, please. The first thing I'm going to teach you about is transcription, how to convert DNA into RNA. The ingredients that you'll need for this is your DNA. You need a big enzyme called RNA polymerase. And you will also need these little things here called RNA bases. Now, with those three starting materials, you're going to be able to take that DNA and read it and convert it to RNA. So what happens is the RNA polymerase binds to the section of DNA you're trying to read. It peels open the two-stranded DNA, and it's going to then start reading the strand of DNA and bringing the RNA bases that are complementary to that DNA strand. As the RNA polymerase chugs along the DNA, it adds more and more bases that are complementary to that DNA strand there. Eventually, you're going to make a long enough strand of RNA, which we will call mRNA, standing for messenger RNA, that will be the coding RNA to build the protein from. Because again, you have to first take the DNA, convert it into an RNA strand, and then you take that RNA strand, and in the next step, we're going to see how we convert it into a protein. So eventually, once the RNA polymerase is done reading the DNA and building the mRNA, it's going to fall apart. The RNA polymerase will go back into the nucleus, and the mRNA will then release itself from the DNA, leaving the DNA unharmed. This mRNA creation process is termed transcription. And transcription is going to be the process of converting DNA to RNA, and it all takes place in the nucleus. Eventually, this mRNA is going to leave the nucleus, and it's going to be converted into a protein. However, before that mRNA leaves the nucleus, it needs to go through a process called mRNA processing. So let's say we have the DNA. We turned the DNA strand into the mRNA strand. So far, this is as much as you've learned. That process is called transcription. Now, the mRNA strand is called an unprocessed mRNA strand. It's like the raw mRNA, not modified at all yet. We have to modify this a little bit because the RNA polymerase literally took every single piece of that DNA and just copied it into the RNA version of it. This whole copy sometimes isn't what you need to build the protein. Sometimes all you need is A, D, E, and G. But you have to transcribe the whole gene first. So in order to get the mRNA to be the sequence that we need for particular proteins, we go through a process called mRNA processing. This, again, happens in the nucleus, and it's the process where we're able to chop up these pieces and bring them together with variety. This whole process of chopping up pieces of mRNA and bringing them together into a variation of sequences is called alternative splicing. Now, if you see the two examples of the two protein sequences we took the unprocessed mRNA, and for protein 1, we chopped out A, C, G, and I. The rest of them stayed together. The parts that we chopped out are called introns because they stay in the nucleus. The parts that we left as our primary mRNA sequence for the protein are called exons because they exit the nucleus. If you're going to build the protein, it happens outside of the nucleus. This example here, same thing. We chopped out some introns to stay in the nucleus, and the coding sequence, which is made up of exons, is going to leave the nucleus to be coded into a protein. So let's do a little bit of review. DNA converted to RNA is called transcription. RNA goes through mRNA processing to be modified, sliced and diced into introns and exons. The exons leave the nucleus and they get bound by ribosomes. So ribosomes are also termed rRNA. And rRNA, or ribosomes, I'll just stick with the word ribosomes, bind onto the mRNA strand. They have this like natural attraction towards each other. You never want mRNA to be swimming freely in your cytoplasm because it's going to be eaten up by some other enzymes. So ribosomes grab onto the mRNA, and this again is all happening in the cytoplasm. When the ribosome grabs onto the mRNA. It's able to read the sequence on the mRNA and start bringing in these structures called tRNA, transfer RNA. And at the tippity top of these tRNA molecules, you have amino acids. Now again, let's review a little bit. The DNA got turned into RNA. This RNA got bound by a ribosome, and it's going to be read by this ribosome, and the proper tRNAs holding on to proper amino acids are going to come into the sequence. Now, what do I mean by proper? Well, the sequence of the mRNA dictates which amino acids get to come in. And I want you guys all to remember that proteins are built out of amino acids. So we bring in the amino acids to bind together to build a long chain that's eventually going to be termed a protein. If we do take a closer look, and this is where the nitty-gritty details are, you see the mRNA sequence here. And it shows you sequence like UUC, UGG. Every 3 bases, let me get my pen here. Every 3 bases we term a codon. So that's a codon, and that's a codon. And I'm going to write the word here. C-O-D-O-N. It is kind of difficult to write with my MacBook because it's not a pen, but you get the idea. It is called a codon. Now the ribosome again is holding onto the mRNA, reading the codons. It's going to bring in the proper tRNA that has the matching anticodon. So if you recall from the last 2 PowerPoints, you have A's matching with T's, but in RNA you don't have a T, you have a U. And you have G's pairing with C's. This is the proper pairing of bases together. So with a U, it's an A. With a U, it's an A. With a C, it's a G. This AAG is the proper anticodon for the codon that's on mRNA. Now the next codon says UGG. The proper anticodon is ACC. Now why do these sequences matter? Well, because the amino acid matters. UUC is the code for phenylalanine. UGG is the code for tryptophan. AAA, which matches up with UUU, this AAA codes for lysine. So the codons on the mRNA will dictate which tRNA comes in, dropping off the proper amino acid. You must have the right amino acid sequence to build a functioning protein. So what we're going to do together is we are going to look at a sequence of DNA, we're going to convert it to RNA, and we're going to convert it to a protein. Before we do that, I do have to introduce to you this chart called a codon chart, and this will be given to you on a test because this will definitely be a test question. A codon chart, if you recall from the last slide, every 3 bases on mRNA is called a codon. So you have UUC or UGG. Let's say we want to look up UUC. So we look at our codon chart. I'm trying to figure out which amino acid does UUC match with. So the first base is a U. So we're going to go up here. We're going to be in this row. The second base is also a U. So UU, we're going to be in this box here. The third base is a C. So we go across and there's UUC. UUC is phenylalanine. So again, write this down. This is a codon chart, not an anticodon chart. Where a lot of students make mistakes is they say, oh look, AAG, and that's what they look up on this chart. That is not what you should be looking up. You should be looking up the codon, UUC. Use the bases that are on the mRNA. The anticodons are there to bind with the codon in order to drop off the proper amino acid. These amino acids, their codes are from the mRNA. So again, you'll be given this on the exam. What we're going to do together now is an example. I've given you the DNA sequence, and I'm going to ask you for the RNA sequence and then the codon sequence. So I'm going to do part of this for you, and then you guys can pause it and see if you can finish it on your own, and then I will go over the answer. If the DNA sequence is TAC, and we're going to write down what these guys pair with. So A's pair with T's, but in RNA, it's not a T, it's a U, and G's pair with C's. That's a little cheat sheet right there. So T in DNA, what does a T pair with? A T pairs with an A. What does the A pair with? Well, the A pairs with a T, but in RNA, do we have T's? Nope, we don't have T's. We have U's. So with that being said, the A is going to pair with a U. The C pairs with a G. So go ahead and complete the rest of this RNA sequence for me. You can pause the video and I will give the answer once you've completed it. So go ahead and pause the video now and try to get the rest of the RNA sequence. All right, so let's go over the answer. GGA will pair with C, CU. TTT will pair with A, A. ATA will pair with UAU. ATG will pair with UAC. GGC, that's an easy one, CCG. CGC will be GCG. And then TTG will be AAC. This is the proper RNA sequence. Now, what you need to do on your own is figure out the codon sequence. The codon sequence will be from the previous slide, and you will use that codon chart in order to figure out what each codon stands for. AUG. I'll just start it off for you, and then I'll give you guys the chance to do it yourself. AUG is your start codon. And now you can go ahead and figure out the rest of this on your own. I will give this answer at our next Zoom session. So be sure that you attend so that I can give you the key to this codon sequence. But use the previous chart to figure out what CCU is, AAA, and so on and so forth. You may simply use the abbreviations. Again, you do not need to memorize the codon chart. I will give it to you. All right, guys. I hope this wasn't too bad. Stay tuned for our next PowerPoint.